Very pleasant morning to you all. Welcome back again to our session of the topic respiration in plants. Last class we had discussed that is what is aerobic respiration. Now we shall discuss anaerobic respiration. Clear? Now anaerobic respiration means what? Taking place in the absence of oxygen. Got it? That's why it is termed as anaerobic. Next, when we talk about anaerobic respiration, glucose, that is sugar, is partially broken down. If you are asked the characteristics, partially broken down. Got it? And the products that we get is either ethyl alcohol in plants, or we say it ethanol or lactic acid. Clear? Now the overall reaction, let me show you. C6H12O6. In the absence of oxygen, it partially breaks down into C2H5OH or ethyl alcohol or ethanol. Ethyl alcohol plus carbon dioxide plus 2 ATP. Clear? Now this carbon dioxide which is produced, along with it, this is, you put an arrow indicating the release of gas, and ATP, adenosine triphosphate. So you can balance it, 2C2H5OH, 2CO2. Got it? Ethyl alcohol is also known as ethanol. Whereas in aerobic respiration, what we had discussed, that glucose or sugar in the presence of oxygen breaks down into carbon dioxide, water and 38 ATP. That's why it is called as oxybiotic. Got it? And it is referred as an oxybiotic. Clear? Sometimes you may be asked the difference between anaerobic and aerobic respiration. The first point of difference, what will you write? In aerobic, glucose is completely broken down into carbon dioxide, water and 38 ATP. Whereas in anaerobic respiration, it is partially broken down into ethyl alcohol or ethanol or lactic acid in animals. Carbon dioxide and the amount of energy is only 2 ATP. Second point of difference, anaerobic respiration takes place in fungi and bacteria. Clear? Whereas certain seeds also can respire anaerobically, but for a limited period. In animals, it takes place in muscle cells. Whereas aerobic respiration takes place in all living cells in higher organism. Clear? That is the main point of difference. Next, you can write that the products produced are ethyl alcohol or lactic acid, whereas the end products are carbon dioxide, water in aerobic respiration. Next point of difference, you can say the amount of energy released in anaerobic is less, 2 ATP, whereas in aerobic respiration, the amount of energy released is more, that is 38 ATP. Clear? These are the main points of differences taking place in, that is between aerobic and anaerobic respiration. Let us now discuss about certain experiments. Got it? To prove that how, that is oxygen is used up in respiration. Clear? Now in this experiment, what we are going to prove, that oxygen is used up in respiration. Clear? Experiments to show the utilization of oxygen in respiration. What we are taking? We are taking two conical flasks. Why they are called conical flasks? Because they are shaped as in a cone, that is triangular shape. So we mark it as A and B. Clear? This is wet cotton wool. We take some amount of wet cotton wool and introduce it at the bottom of the two conical flasks. Clear? Next, what do? We place some seeds, germinating seeds, that is pea seeds or gram seeds, in one conical flask A, marked A, and in the other we take boiled seeds, that is pea seeds. If you are taking pea seeds, here also you have to take pea seeds. If you are taking gram seeds, you have to take your gram seeds. Now, care should be taken that equal number of seeds are being taken. Like suppose you are taking 5 to 6 seeds, here also you require 5 to 6 seeds. Clear? 1, 2, 3, 4, 5. So 5 seeds here, 5 seeds here. What is the difference? In flask A, the seeds are germinating seeds, and in flask B, the seeds are first boiled so that the cells are killed. Now, before introducing into this flask B, the boiled seeds, pea seeds, should be treated with, that is antiseptic, that is chemical such as formalin or carbolic acid. What is the advantage? To prevent the seeds from bacterial decay. Clear? Next, we introduce a small test tube. Got it? Containing soda lime. What is soda lime? It is a mixture of sodium hydroxide and silicate lime. Clear? What is the purpose of soda lime? To absorb any carbon dioxide. Clear? And then you have a bent conical, that is tube, you can say, which passes out from each of the flasks and then introduced into a beaker containing water. This is the setup. Setup A is called as experimental setup. Setup B is called as control setup. What is a control setup? It is a setup in which all the conditions are same except the one which we want to study. Clear? Now, what do we observe? We observe that there is a rise in the level of water in this delivery tube. These two are the delivery tube and this is the cork. Clear? Now, what we do find? Water rises in this delivery tube up to a certain level, whereas the rise of water in the delivery tube passing from flask B is less. Why is it so? Because the seeds, they have used up oxygen which is present inside the conical flasks. Now, you know that in the atmosphere, the amount of oxygen is 21%. Clear? So, when this oxygen inside the flasks is used up by the germinating seeds, that is compensated by the rise in level of water. Clear? Whereas the CO2 is already absorbed by the soda lime. And in flask B, there is no rise in the level of water because their seeds are dead seeds. They are already boiled, so the cells are killed. Clear? So you find that there is very little rise in level of water. So this proves that the seeds, that is the germinating seeds, or during respiration, oxygen is required. Oxygen is used up during respiration. Clear? So, if you are given this setup, what type of questions you may be asked? You may be asked that what is the function of soda lime? The function of soda lime is to absorb any carbon dioxide released during respiration. Clear? Now, second question you may be asked, like why the seeds are being boiled and then treated with carbolic acid or formalin to prevent bacterial decay. Then you may be asked, write the definition of aerobic respiration or simply respiration. You may be also asked why there is rise in the level of water in the delivery tube from coming out from flask A. That is, you have to say the answer. It is because the oxygen which is used up during respiration creates a vacuum which is compassionated by the rise in the level of water. Then you may be asked, what do you mean by control setup? It is a setup in which all the conditions are same except the one which we want to study. So what do we want to prove in this experiment? That oxygen is used up in aerobic respiration, or in other words, in aerobic respiration, it takes place in the presence of oxygen. Clear? Now come to the next experiment. You write this definition of control. Sometimes it may be asked here. You already have the book there. The setup in which the conditions under study is missing or is called the control, or you can say in your own words, it has all the conditions same as the experimental setup except the one which we want to study. Now come to the next experiment. Experiment to prove that carbon dioxide is produced during respiration in germinating seeds. Now see, here what do we take? We take two round bottom flasks. Clear? Why it is called as round bottom? Because the lower portion is round. Again you place some wet cotton wool to provide moisture, because for germination water or moisture is required. You place some germinating seeds in experimental setup A and boiled seeds in the setup B. The boiled seeds before, that is the gram seeds, you first treat it with carbolic acid or formalin to prevent bacterial decay. Now what happens during the process of respiration? Carbon dioxide is produced. It gets collected in the flasks, that is the round bottom flasks. Now what do? You prepare a freshly prepared lime water. How it is prepared? Take a glass of water, squeeze a lemon or lime in it, and then strain it. Take some amount of it in a test tube. This is your freshly prepared lime water. Now what happens? After few hours, the carbon dioxide which gets collected in the flasks due to respiration, now tilt the round bottom flasks into the lime water, that is the test tube containing lime water. What do we find? The clear lime water turns milky, proving that CO2 or carbon dioxide is produced during respiration. Whereas this setup B or the control setup, when it is tilted into another flask having freshly prepared lime water, what do we find? The lime water does not turn milky, but even if it turns, it takes a very long time because some CO2 is already present here. But what does it prove in this experiment? That carbon dioxide is produced during respiration. Clear? So again you may be asked the same question, that is how to prepare lime water, why do we treat the boiled or the dead seeds with formalin, got it? And the overall reaction of aerobic respiration. So that is all for today. Stay home, be safe.